Welcome to the Deep Dive. We're here to do more than just skim the surface. We aim to map out knowledge, connect the crucial ideas, and give you that integrated understanding needed to really master a subject.
And today, well, it's a big one. We're tackling biology.
Yeah, the scientific study of life, it's incredibly vast, isn't it? I mean, it covers everything from tiny molecules all the way up to the entire planet, the biosphere.
It really does. And if you try to approach all that information without some kind of structure, well, you're just going to get lost. It's overwhelming. So our goal today is to provide that structure.
Exactly. Our mission is to take this huge subject and organize it into a framework that makes sense, one that's comprehensive and, maybe most importantly, interconnected. We're talking about the 5 core unifying themes of biology.
Think of it as the blueprint, maybe. The plan that shows how every single piece of biological knowledge, whether it's genetics or ecology or climate change, actually fits together.
Right. And to show how this framework works right from the start, let's bring in our central example. It's a little creature called the beach mouse, Paramiscus polionotus.
Ah, yes, the beach mouse, a fantastic illustration.
So picture this. You've got 2 populations of this exact same mouse species. They live just a few miles apart in Florida. One group lives right on the coast on these bright white sand dunes, and their fur, it's very light, almost white, perfect camouflage, really.
Blends right in.
Then you go just a little bit inland, and there's another population living on much darker, fertile soil, and their fur is significantly darker.
And that difference, that simple difference in coat color,
Yeah.
it seems minor, but it's actually where all 5 of our themes intersect. It's quite remarkable.
How so? Let's break that down.
Okay, well, the color itself, that's an inherited trait, right?
Okay.
So that points directly to genetic information theme 2.
Got it. Information encoded in their genes.
Exactly. Then the biological machinery, the cells and molecules needed to actually produce that fur color, that requires intricate organization, which is theme 1.
Okay, organization within the mouse's body.
Then, of course, the mouse needs fuel to live, to grow, to reproduce. That brings in theme 3, energy and matter.
Right, the basics of survival.
And its survival, especially with that coat color, is heavily influenced by its interactions, theme 4. Interactions with predators, interactions with its specific environment. The color only matters because of those interactions.
The camouflage is only an advantage because there are predators looking for them.
Precisely.
Yeah.
And finally, the reason for the difference between the 2 populations, why one is light and one is dark, that's fundamentally explained by evolution, theme 5, the core theme.
Wow. Okay, so that one little difference really does touch on everything. That's the 5-point framework we're diving into today. We want to extract that, professional level insight from each one. Let's list them out clearly again.
Okay. 1, organization.
2, information.
3, energy and matter.
4, interactions.
And 5, the big one, evolution.
All right, let's start with theme 1, organization. This really begins with understanding that life isn't random. It's structured in a very specific hierarchy. You can't really talk about life without talking about its structure, and structure is always linked to function.
That's right. And we can map this hierarchy across 10 distinct levels. Let's start really broad at the global scale and zoom our way down.
Okay, where do we begin?
We start with the biosphere. This is literally everything, all life on Earth and every single place life exists, from the bottom of the ocean to high up in the atmosphere.
The whole living planet. Got it. Zooming in.
We hit an ecosystem. Now, this is more localized. It includes all the living things in a particular area, say a mountain meadow, plus the non-living components they interact with, things like soil, water, sunlight, temperature.
Living and non-living together in one functional unit.
Okay,
Next level down.
that would be the community. This is just focusing on the living parts within that ecosystem. So it's the whole array of different species, all the plants, animals, fungi, microorganisms that live and interact in that meadow.
Okay, so just the life forms in that ecosystem and within that community.
We find specific populations. A population is defined as all the individuals of one particular species that live within a defined area and, crucially, have the potential to interbreed. So our group of beach mice on that particular stretch of dunes is a population.
All right, a group of the same species. And then finally, the individual.
Exactly. The organism, the single living thing, like one specific mouse or one lupine plant in that meadow.
Okay, so that takes us from the global down to the individual. But the organization continues inside the organism, right?
Oh, absolutely. An organism like a plant is made up of organs. A leaf, for example, is an organ designed for photosynthesis. A root is an organ for water absorption.
And organs are made of.
Tissues. A tissue is a group of similar cells working together to perform a specific function, like the epidermis, the outer protective tissue of a leaf, or the mesophyll tissue inside, which is packed with photosynthetic cells.
Which brings us to the cell. You called this the critical juncture earlier.
It really is. The cell is the fundamental unit of life. All structure, all function, ultimately traces back to the activity of cells. Everything an organism does is based on what its cells are doing.
But we can go even smaller, inside the cell.
Yes. Inside the cell, we find organelles. These are the functional components, often enclosed by their own membranes in more complex cells. Think of the nucleus holding the DNA, or the chloroplasts where photosynthesis actually happens. They're like the cell's own specialized organs.
And the final level, the base level.
That's the molecule. A chemical structure made of two or more atoms bonded together. Water is a molecule. DNA is a giant molecule. Chlorophyll, the pigment that captures light, is a molecule.
Okay, 10 levels. Biosphere, ecosystem, community, population, organism, organ, tissue, cell, organelle, molecule. Phew. But it's not just a list, is it? You mentioned something about emergent properties.
Right, this is absolutely key. This hierarchy isn't just about scale, it's about how complexity arises. New properties emerge at each step up that are absent from the level below.
How does that work? Don't scientists usually try to understand things by breaking them down? Reductionism.
They do, and reductionism is incredibly powerful. Reducing complex systems to simpler parts, like isolating DNA to figure out its double helix structure, has been essential. But it has limits. It can't explain everything.
Because of these emergent properties.
Exactly. An emergent property is something new that arises from the specific arrangement and interaction of parts at a lower level. My favorite example is photosynthesis. You can take all the molecules needed, the chlorophyll, enzymes, etc., out of a chloroplast and mix them in a test tube.
And nothing happens.
Nothing. Photosynthesis, the ability to convert light energy to chemical energy, only emerges when those molecules are arranged in a very specific, highly organized way within the intact chloroplast structure. The organization itself creates the function.
Ah, okay. So the whole is literally more than the sum of its parts because of the way the parts are put together.
Precisely. Think about consciousness. You can study individual brain cells, neurons, and the chemical signals between them. But thought, memory, consciousness itself, those are emergent properties arising from the incredibly complex interactions of billions of those neurons organized in a specific way. Reductionism alone can't explain it.
Which leads to
Systems biology. This is the necessary partner to reductionism. Instead of just looking at parts in isolation, systems biology focuses on analyzing the interactions among those parts. How do networks of genes regulate each other? How do feedback loops between hormones create a stable internal environment? How does a change in one species affect the entire food web?
It's about understanding the connections, the network, the system as a whole.
Exactly. It's about modeling the dynamic behavior of the entire biological system. For example, figuring out how all the molecular interactions in our cells work together to produce our 24-hour sleep-wake cycle. That's systems biology.
And underlying all of this, at every single one of those 10 levels, is this principle you mentioned, structure fits function.
Always. It's one of the most fundamental observations in biology. The shape of something is directly related to what it does. Think about a bird's wing. Its lightweight structure with feathers is perfectly adapted for flight. Or a leaf, broad and flat to maximize sunlight capture.
Or even that hummingbird you mentioned in the outline, with its unique shoulder joint allowing it to hover and fly backwards.
Perfect example. That specific structure enables a unique function. And this match isn't an accident. It's the result of evolution shaping organisms over time.
Okay, let's zoom back into that crucial level.
Yeah.
The cell. You said it's the fundamental unit.
It is. Cell theory, which dates back to the 1800s, established this. Every action an organism takes, from you moving your eyes to read this, which involves muscle and nerve cells, to a tree absorbing water through root cells, is based on the activities of cells.
And all cells have some common features, like a boundary.
Yes, every single cell is enclosed by a membrane, a plasma membrane, that acts as a selective barrier, controlling what goes in and out. But beyond that basic similarity, cells diverge into two fundamentally different structural types. This is a major split in the history of life.
What are the two types?
First, you have prokaryotic cells. These are found in the domains bacteria and archaea. Their defining feature is, well, their relative simplicity. They lack a true nucleus to contain their DNA, and they generally don't have other internal compartments enclosed by membranes, what we call organelles. They're usually smaller too.
So simpler, smaller, no nucleus. That's prokaryotic. The other type.
The eukaryotic cells. These make up all other forms of life, plants, animals, fungi, and protists. They do have a true nucleus, a membrane-bound sac that houses the cell's DNA. And they also contain numerous other specialized membrane-enclosed organelles.
Like the chloroplasts for photosynthesis we mentioned.
It's exactly. Or mitochondria for energy processing, the endoplasmic reticulum for making proteins and lipids. Lots of internal compartments. This compartmentalization allows eukaryotic cells to perform many different complex chemical reactions simultaneously in different locations without them interfering with each other. It enables a much higher degree of complexity and specialization.
Which makes sense why they form the basis of larger multicellular organisms like us.
It's a key factor. That internal organization allows for the specialization needed to build complex tissues, organs, and ultimately entire organisms.
Okay, so we have this intricate organization, especially in eukaryotic cells, but none of that structure works without instructions, which brings us neatly to theme 2, information.
Right. The master blueprint for life is DNA, deoxyribonucleic acid. This giant molecule is organized into structures called chromosomes, usually found within the nucleus in eukaryotes.
And specific parts of that DNA are the genes.
Correct. Genes are the functional units of inheritance. They are specific segments of DNA that contain the instructions, the encoded information needed to build cellular molecules, mostly proteins, which then carry out the cell's functions.
How does DNA actually store so much information?
It's down to its molecular structure. DNA is famous for being a double helix, two long strands twisted around each other. Each strand is a chain made up of 4 chemical building blocks called nucleotides.
Adenine, thymine, cytosine, and guanine, A, T, C, and G.
Those are the ones. The sequence, the specific order of these 4 letters along a DNA strand, is what encodes the genetic information. It's like a 4-letter alphabet that can write incredibly complex instruction manuals.
So the difference between, say, a mouse gene and a human gene is just the order of those A's, T's, C's, and G's.
Fundamentally, yes. The sequence dictates everything, but having the information stored isn't enough. The cell needs to use it. That's where gene expression comes in.
Right, making the blueprint actually do something. How does that process work? You mentioned the central dogma.
Yes. The central dogma of molecular biology describes the basic flow of genetic information. It typically starts with transcription. Here, the sequence of a specific DNA gene is accurately copied into a different nucleic acid molecule called messenger RNA, or mRNA.
So DNA gets transcribed into mRNA, like making a working copy of one page from the master blueprint.
That's a great analogy. The mRNA molecule then travels out of the nucleus in eukaryotes to the main part of the cell.
And then what happens to the mRNA?
Next comes translation. The sequence of nucleotides on the mRNA molecule is read by cellular machinery called ribosomes. They read it in 3-letter words called codons. And each codon specifies a particular amino acid, which are the building blocks of proteins.
So the mRNA sequence gets translated into a chain of amino acids.
Exactly, a linked chain. But just having a chain of amino acids isn't usually functional. The final critical step is protein folding. That linear chain of amino acids has to spontaneously fold up into a very specific, unique 3-dimensional shape. It's the information in the amino acid sequence itself that dictates how it folds, and it's that final 3D shape, the structure, that determines the protein's function.
Ah, back to structure fits function. So if it folds wrong,
It usually won't work, or worse, it could be harmful. Think about the crystalline protein in the lens of your eye. It has to fold perfectly to be transparent and help focus light. If it misfolds, you get cataracts.
wow, okay. Transcription, DNA to mRNA. Translation, mRNA to amino acid chain. Protein folding, chain to functional shape.
Got it.
It.
And what's truly remarkable, providing incredibly strong evidence for the shared ancestry of all life, is the universality of the genetic code. Meaning that the code, the dictionary that translates mRNA codons into amino acids, is essentially the same across all known life forms. The codon AUG means the amino acid methionine, whether you're looking at a bacterium, a mushroom, a tree, or a human.
The same genetic language for everyone. That's powerful evidence for a common ancestor.
It really is. Now, understanding all this information, especially across entire organisms, has exploded in recent decades with the fields of genomics and proteomics.
Okay, what's the difference there?
The genome is the entire set of genetic instructions for an organism, all of its DNA. For humans, that's roughly 3 billion nucleotide pairs in each set of chromosomes. Genomics is the study of whole genomes, often comparing genomes between species or individuals. It applies that systems biology approach we talked about, but at the level of genes and DNA.
So looking at the whole library, not just one book, and proteomics.
Proteomics is the parallel study of the entire set of proteins produced by an organism or cell that's called a proteome. It looks at not just which proteins are present, but their quantities, their modifications, and crucially how they interact with each other.
Because the proteins are the actual workers in the cell, carrying out the instructions from the genes.
Exactly. Studying the proteome gives you a more direct picture of what's actually happening functionally. But analyzing genomes and proteomes generates unbelievable amounts of data. This was only possible because of three key technological and scientific shifts.
What were they?
First, high-throughput technology. These are automated methods that allow researchers to analyze thousands or millions of samples incredibly quickly. Think robotic sequencing machines that can read DNA bases far faster than any human could.
So generating the raw data much, much faster.
Right. Second, bioinformatics. This is the crucial field that uses computer science, statistics, and mathematics to store, organize, analyze, and make sense of these massive biological data sets. Without powerful computers and clever algorithms, the data from genomics would just be noise.
You need the tools to find the patterns in the flood of information.
Absolutely. And third, the rise of interdisciplinary research teams. Big biological questions now require collaboration between biologists, computer scientists, engineers, mathematicians, chemists, physicists, even ethicists. No single person can master it all.
It's become a team sport, really, bringing different expertise together to tackle these huge, complex data sets and questions.
That's the reality of modern biology at the cutting edge. It's integrated, it's data-intensive, and it's focused on systems.
Okay, so we've covered the intricate organization of life and the complex information systems that direct it. Now let's move on to themes 3 and 4, which deal with the dynamics, how life is fueled, and how its parts interact.
Right. Theme 3 is about energy and matter. And it's vital to understand that these two things behave very differently within an ecosystem.
How so? Let's start with energy.
Okay, energy flow is essentially a one-way street. For almost all ecosystems on Earth, energy enters primarily as light energy from the sun.
Sunlight, the ultimate source.
Yes. Then, organisms called producers, mostly plants, algae, some bacteria, capture that light energy through photosynthesis. They convert it into chemical energy, storing it in the bonds of sugar molecules and other organic compounds they build.
Okay, producers capture light energy, store it as chemical energy. Then what?
That chemical energy is then transferred to consumers when they eat the producers or when they eat other consumers. So energy flows up the food chain.
But you said it's one way. Does it get lost?
It does, in a sense. Every time energy is used to do work like a muscle contracting, a cell dividing, sending a nerve impulse, some of that energy is converted into heat. And that heat is eventually lost to the surroundings. It dissipates. It doesn't get cycled back to be reused by organisms in the same way.
So energy flows through an ecosystem, enters as light, gets converted and transferred, and exits as heat.
Precisely. A constant input of energy, usually from the sun, is required to sustain life.
Okay, so that's energy. How is matter different?
Chemical cycling, or matter cycling, is fundamentally different because matter is recycled within an ecosystem. The atoms that make up organisms, carbon, nitrogen, phosphorus, water, are not lost. They cycle continuously.
How does that cycle work?
Well, plants, for example, absorb chemical elements from the soil and air, incorporating them into their own structures. When a consumer eats the plant, those chemicals are transferred.
Okay, passed along the food chain, like energy. But what happens when organisms die?
That's where decomposers come in. Organisms like bacteria and fungi break down dead organic material, dead plants, dead animals, waste products. In doing so, they return those essential chemical elements back into the soil, water, or atmosphere.
Ah, so the decomposers complete the loop. They make the chemicals available again for the producers to take up.
Exactly. So unlike energy which flows through, matter cycles within the ecosystem. The same atoms are used over and over again. The carbon atom in your breakfast might have been part of a dinosaur millions of years ago.
That's a mind-bending thought. Okay, so one-way flow for energy, cyclical recycling for matter. That brings us to theme 4. Interactions.
Right. Life isn't static. It's defined by interactions at every level. Starting right down at the molecular level, cells rely heavily on feedback regulation to control their own processes.
Feedback. Like getting comments on your work.
Sort of, but it's the output of a biological process influencing that same process. The most common type is negative feedback.
Negative sounds bad, but you said it's most common.
It's essential for stability, for maintaining homeostasis. In negative feedback, the result or product of a process actually inhibits or slows down that same process.
Okay, can you give an example?
Sure. Think about how your body regulates blood sugar. After you eat, your blood glucose level rises. This stimulates your pancreas to release the hormone insulin. Insulin then signals your body cells to take up glucose from the blood and tells your liver to store excess glucose.
So insulin lowers blood sugar.
Right. And as the blood sugar level drops back to normal, that drop removes the original stimulus for insulin release. The pancreas stops secreting so much insulin. The response, lowered glucose, shuts off the pathway that caused it.
I see. The output dampens the initial trigger. It keeps things in balance, like a thermostat controlling room temperature.
Exactly like a thermostat. That's negative feedback, and it's everywhere in biology.
Was there positive feedback too?
Yes, positive feedback is less common, but important in specific situations. Here, the end product of a process actually speeds up its own production. It acts like an amplifier.
An amplifier, example.
The classic example is blood clotting. When you get a cut, platelets start to gather at the site of injury. As they aggregate, they release chemicals. These chemicals attract more platelets to the site, which release more chemicals, attracting even more platelets.
So it snowballs, accelerating the clot formation until the bleeding stops.
Precisely. Another example is the hormone oxytocin during childbirth, where contractions stimulate more oxytocin release, which causes stronger contractions, and so on. Positive feedback pushes a process to completion quickly.
Okay, so feedback regulates things within organisms. What about interactions between organisms?
Absolutely crucial. Ecosystem interactions are the fabric of life. Every organism constantly interacts with other organisms. Think about predator-prey relationships, the hawk hunting the mouse. That shapes the populations of both species.
Or mutually beneficial relationships.
Yes, mutualism, like the cleaner fish that remove parasites from larger fish, benefiting both. Or competition, where different species or individuals of the same species compete for limited resources like food, water, or nesting sites.
You mentioned the African acacia tree in the outline. How does that illustrate these interactions?
Well, acacia tree interacts with so many things. It might have a mutualistic relationship with nitrogen-fixing bacteria in its roots, and maybe with ants that defend it from herbivores in exchange for nectar. It interacts antagonistically with giraffes that eat its leaves. Its seeds might be dispersed by animals that eat its fruit. And when parts of it die, it interacts with decomposers that recycle its nutrients back into the soil.
It's connected in a whole web of interaction.
It's a very complex web. No organism exists in isolation. And they don't just interact with other living things, they constantly interact with the non-living physical environment too. Plants are a great example. They take carbon dioxide out of the atmosphere and release oxygen into it during photosynthesis. Their roots physically break up rocks and release chemicals that contribute to soil formation. Animals breathe oxygen and release CO2. It's a two-way street. Organisms shape their environment, and the environment shapes the organisms.
Which leads us, unfortunately, to the impact of human interactions on the environment at a global scale, specifically climate change.
Yes, this is perhaps the most profound environmental interaction currently underway. Since the Industrial Revolution, humans burning fossil fuels, coal, oil, natural gas, have released enormous quantities of greenhouse gases, especially carbon dioxide, CO2, into the atmosphere.
And these gases trap heat.
They do. They act like a blanket around the Earth, trapping heat that would otherwise radiate back into space. This is leading to a measurable increase in the average global temperature. We've seen about a 1 degree Celsius rise since 1900.
And it's projected to rise more.
Current projections, depending on future emissions, suggest at least another 3 degrees Celsius rise, maybe more, by the end of this century. This overall warming trend and the associated changes in weather patterns, sea levels, and extreme events is what we call climate change. We define it as a directional change in climate lasting 3 decades or more.
What are the biological consequences? We hear about polar bears.
The polar bear losing its sea ice hunting platform is a very direct and visible impact. But the effects are widespread and sometimes more subtle, yet just as devastating. Take the Celloporus lizard studied in Mexico.
What happened to them?
Researchers found that because average temperatures have risen, these lizards are forced to spend much more time hiding in cooler refuges during the day to avoid overheating.
Okay, so they survive the heat by hiding. What's the problem?
The problem is the trade-off. While they're hiding, they're not foraging for food, and they're not engaging in reproductive activities. Less food means lower energy reserves. Less mating means fewer offspring.
Ah, so the indirect effect of the temperature forces behavioral changes that hurt their survival and reproduction in the long run.
Exactly. And detailed studies tracking these populations since 1975 have shown a significant decline. About 12% of local populations have vanished. Not necessarily because they cooked to death, but because the changing climate disrupted the delicate balance of their energy budget and life cycle. It shows how tightly interactions with the environment are linked to survival and ultimately to evolution.
And that brings us directly to theme 5, the absolute core theme that integrates everything else: evolution.
Indeed, evolution is the central organizing principle of all biology. It's defined as the process of biological change over time, where species accumulate differences from their ancestors as they adapt to different environments.
It explains both the similarities and the differences we see in life, doesn't it?
Perfectly. Evolution accounts for the remarkable unity of life, the shared features that point to descent from common ancestors. Things like the basic skeletal plan of vertebrates, one upper arm bone, two forearm bones, wrist bones, finger bones, being present in humans, cats, whales, and bats, despite their very different functions, or that universal genetic code we talked about.
Unity through shared ancestry. But it also explains the flip side.
Yes, the incredible diversity of life. All the differences that have accumulated after lineages diverged from those common ancestors. As populations adapted to countless different ways of life, biologists have currently named about 1.8 million species.
1.8 million.
And that includes maybe a million species of insects alone. But the estimates for the total number of species on Earth, including those not yet discovered, range much higher, maybe 10 million, maybe even 100 million or more. The diversity is just staggering.
How do biologists even begin to organize that kind of diversity?
Through classification, which aims to reflect evolutionary relationships. The broadest level of classification groups all life into 3 large domains.
Okay, what are the 3 domains?
First, domain bacteria. These are prokaryotes, incredibly diverse and widespread. You find them everywhere. There are likely multiple kingdoms within bacteria.
Domain bacteria, prokaryotes. Got it. Second.
Domain archaea. These are also prokaryotes, but they are evolutionarily distinct from bacteria. Many archaea are famous for living in extreme environments, volcanic hot springs, super salty water, deep-sea vents, though they're found in more moderate places too. Again, multiple kingdoms here.
Domain archaea, also prokaryotes, often extremophiles. And the third
Domain eukarya. This includes all organisms whose cells are eukaryotic cells with a true nucleus and organelles. This is where we find the more familiar large multicellular life forms.
And eukarya is further divided.
Traditionally, it was divided into 4 main kingdoms, though the picture with protists is more complex now. But the key groups are Kingdom Plantae, multicellular eukaryotes that carry out photosynthesis like trees and flowers.
Plants, okay.
Kingdom Fungi, which absorb nutrients from their surroundings, often after decomposing dead organic matter like mushrooms and yeast.
Fungi, got it.
Kingdom Animalia, multicellular eukaryotes that ingest other organisms for food, like insects, fish, mammals,
Animals. And the last group.
the protists. This used to be a single kingdom, but it was kind of a catch-all category for eukaryotes that weren't plants, animals, or fungi. It includes mostly single-celled organisms like amoebas and paramecia, but also some multicellular forms like kelp. We now know it's not a single evolutionary group, and scientists are classifying protists into several different groups to better reflect their true evolutionary relationships based on genetic data.
Okay, so bacteria, archaea, and eukarya, containing plants, fungi, animals, and various protist groups. That's the basic map of life's diversity. Now, how did Charles Darwin explain how this diversity arose?
Darwin's genius, published in his book On the Origin of Species in 1859, was built on 2 main points.
What was the first?
The first was descent with modification. This concept captures that duality we just discussed. Descent implies shared ancestry, the unity of life. Modification implies that as descendants moved into different habitats over millions of years, they accumulated changes or adaptations, leading to the diversity we see.
So, unity and diversity explained by common ancestry plus accumulated changes. What was his second big idea?
The second, and arguably the most revolutionary, was the mechanism that drives most of that modification: natural selection.
Natural selection. How did he arrive at that?
Through incredibly careful observation and logical inference, he put together several facts that were largely already known.
What were his key observations?
He made 3 critical ones. First, heritable variation. Individuals within any population vary in their traits, like the different coat colors in mice, and many of these traits are passed down from parents to offspring. That links back to theme 2, information.
Okay, variation exists and it's inherited. Observation 2.
Overproduction and competition. All species have the potential to produce far more offspring than the environment can possibly support with resources like food and space. This leads inevitably to a struggle for existence or competition.
More offspring are born than can survive. Makes sense. And the third observation.
Environmental adaptation. Species generally seem well suited to their specific environments. Birds in seed-rich areas have strong beaks for cracking seeds. Animals in cold climates have thick fur. The beach mice match their sand. The inland mice match their soil.
Okay, variation, overproduction, and adaptation. How did he connect those dots?
Through logical inference, he reasoned. If there's variation, and if more individuals are produced than survive, then individuals whose inherited traits give them a higher probability of surviving and reproducing in that specific environment will tend to leave more offspring than other individuals.
So the ones better suited to the local conditions have more kids, basically.
Essentially, yes. And if this unequal reproductive success continues over many generations, the result will be adaptation. Favorable traits will accumulate in the population because the environment is, in a metaphorical sense, selecting for the propagation of those traits.
So natural selection isn't a conscious force, it's just the outcome of differential survival and reproduction based on how well an organism's traits fit its current environment.
Precisely. Darwin called it an editing process. It doesn't create new traits out of thin air. It acts on the existing variation within a population, favoring some traits over others in a specific context. The batwing didn't appear magically. It's a modification of the standard mammalian forelimb, heavily selected for because it conferred the advantage of flight in the ancestors of bats.
And this process, repeated over vast stretches of time, branching out in different environments, explains the entire tree of life.
That's the idea. The tree of life is a powerful metaphor and model. Species that are closely related, like the different Galapagos finches, share a recent common ancestor. If you trace lineages back far enough past the split between animals and plants, past the origin of eukaryotic cells, you eventually reach the earliest prokaryotic ancestors, over 3.5 billion years ago, connecting all known life forms.
Let's bring it back to those specific examples, the Galapagos finches.
Classic example of adaptive radiation.
Oh.
Darwin proposed that an ancestral finch population reached the Galapagos Islands, and as different groups became isolated on different islands with different food sources, natural selection favored different beak shapes.
Right, you mentioned heavy beaks for seeds on one island, slender beaks for insects on another. The environment selected the beak structure that worked best locally.
Exactly. The available food source was the selective pressure driving the modification of the ancestral finch beak.
And our beach mouse, how does it fit this perfectly?
It's a microcosm of the whole process happening right now. You have the heritable variation, coat color, theme 2. You have the selective pressure from the environment, visual predators like hawks and owls, theme 4. The mice, whose coat color provides better camouflage in their specific habitat, light on beach, dark inland, are less likely to be eaten and thus more likely to survive and reproduce.
So over generations, selection maintains the light coat color in the beach population and the dark coat color in the inland population. It's adaptation driven by predation.
Precisely. It perfectly illustrates how evolution, driven by natural selection acting on heritable variation in the context of environmental interactions, leads to adaptation. It ties themes 2, 4, and 5 together beautifully.
Okay, we've established evolution as the core theme, built upon organization, information, energy matter, and interactions. But how do we know all this? That brings us to the process of science itself. Section 4, Scientific Inquiry and Collaboration.
Right. All this biological knowledge, especially a powerful theory like evolution by natural selection, wasn't just dreamed up. It's built on a rigorous process. Scientific inquiry. Science isn't just a body of facts, it's a way of knowing. It's a dynamic process of asking questions, seeking information, and finding explanations about the natural world.
And it often starts with observation, right? Collecting data.
Absolutely. Data are recorded observations, the raw material of science. This data can be qualitative, meaning descriptive. Think of Jane Goodall's detailed field notes on chimpanzee behavior descriptions, the social interactions, tool use, etc.
Okay, descriptive data. What's the other type?
Quantitative data. This involves numerical measurements, counting the number of mice caught, measuring the length of finch beaks, recording changes in CO2 levels. Quantitative data can, and usually must, be analyzed using statistics to determine if patterns are significant or just due to chance.
So scientists collect qualitative or quantitative data. How do they use it to build understanding? You mentioned two types of reasoning.
Yes. Scientific logic often employs both inductive and deductive reasoning. Inductive reasoning is about going from specific observations to broader generalizations. Like observing that every living organism examined so far, from bacteria to elephants, is made of cells, leading to the general principle. All organisms are composed of cells. You induce a general rule from many specific instances.
Okay, specific to general, what about deductive reasoning?
Deductive reasoning flows in the opposite direction from general premises down to specific expected results. This is the logic typically used to test hypotheses. It often takes the form of if-then statements.
Give me an example.
Okay, if the camouflage hypothesis for the beach mice is correct, general premise, then we should predict that non-camouflaged mouse models placed in their habitat will be attacked by predators more often than camouflaged models. Specific expected result. Deduction allows you to generate testable predictions from a hypothesis.
That leads perfectly into the difference between a hypothesis and a theory. People often use theory very casually, like, oh, I have a theory about why my car won't start. But in science, it's much more rigorous, isn't it?
Vastly more rigorous. This distinction is crucial for professional understanding. A hypothesis is a tentative, testable explanation for a specific observation or question. It's an educated guess based on available data, and it must lead to predictions that can be tested by experiment or further observation. My car won't start because the battery is dead is a testable hypothesis.
Okay, a specific testable guess. How is a theory different?
The scientific theory is fundamentally different in 3 key ways. First, scope. A theory is much, much broader than a hypothesis. It explains a wide range of observations and phenomena. The theory of evolution by natural selection explains everything from antibiotic resistance in bacteria to the fossil record to the structure of DNA.
So much bigger picture. Second difference.
Predictive power and testability. A good theory is so general and well-established that it spins off many new, specific, testable hypotheses. Darwin's theory generated countless hypotheses about adaptation, speciation, genetic inheritance, etc., that researchers have been testing for over 150 years.
Okay, it generates more science. And the third, maybe most important difference.
Evidentiary support. This is critical. A scientific theory is supported by a massive, diverse, and continually growing body of evidence from many different lines of inquiry. It has withstood repeated testing and has not been contradicted by any credible scientific data. It's as close to scientific fact as we can get for such a broad, explanatory framework. It's not just a guess. It's a robust, well-supported explanation.
That's a really important distinction. And science also has limits, right? It can't answer every question.
Correct. The key limitation stems from the requirement that hypotheses must be testable and falsifiable. Science deals only with natural explanations for natural phenomena. Questions about the supernatural, about religious faith, about ultimate purpose or morality, these are outside the realm of science because they cannot be investigated using the scientific method of empirical testing and observation. Science can't prove or disprove the existence of ghosts, for example.
It sticks to what can be observed and tested in the natural world. Let's see that testing in action with the beach mouse experiment you mentioned earlier, the camouflage hypothesis.
Right. Testing the hypothesis that coat color provides camouflage protection against predators required a controlled experiment. Biologist Hopi Huckstra and her team did a really elegant one back in 2010.
Remind us of the setup.
Their hypothesis was clear. Mice, whose coat color doesn't match their background, will be preyed upon more heavily. To test this, they made hundreds of plastic mouse models, completely realistic replicas, but made of plastic. They painted some light, matching the beach fan, and some dark, matching the inland soil.
Okay, artificial mice. What did they do with them?
They placed these models out in both habitats, the beach and the inland area. In each location, they put out both light and dark models, scattered randomly.
So how was it controlled?
The control group in each habitat consisted of the models that were camouflaged, the light models on the light beach sand, and the dark models on the dark inland soil. The experimental group consisted of the models that were not camouflaged, the dark models on the beach, and the light models inland.
Ah, okay, so the only difference between the control and experimental groups in each location was the color contrast against the background.
Exactly. The independent variable they were manipulating was the match between coat color and background. The dependent variable they measured was the outcome, the percentage of models that showed signs of being attacked by predators, like bite marks or peck marks.
And the results?
The results strongly supported the hypothesis. In both habitats, the non-camouflage models were attacked far more frequently than the camouflage models. Clear evidence that camouflage significantly reduces predation risk.
And you mentioned the controlled part doesn't mean controlling everything.
Right. You can't control the weather or exactly which predators show up, but by using a control group that experiences all those same factors, except for the variable you're testing, the color mismatch, you can isolate the effect of that specific variable. That's the power of a controlled experiment.
And this built on earlier work.
Yes. Science rarely happens in a vacuum. Hoekstra's work built on earlier studies, including a fascinating one by D.W. Kaufman in 1974. He also tested predation on light versus dark mice, but he added another environmental factor: moonlight.
Moonlight, how did that affect things?
Kaufman used live mice, light and dark, in large outdoor enclosures with a barn owl predator. He ran trials on both light and dark soil, and critically, under both full moon and new moon, no moonlight conditions.
What did he find?
He found that the owl's hunting success was dramatically higher under the full moon, especially when the mouse contrasted sharply with the soil. The worst-case scenario for a mouse was being a dark mouse on light soil. During a full moon, they were easily spotted. It showed how environmental factors, like light levels, interact with the organism's traits, color, to influence survival.
So Hoekstra refined the question, focusing just on color contrast with the models, building on Kaufman's insights about the importance of visual predation.
Exactly. Science is cumulative, building on previous discoveries.
Which brings us to the final part of this section, science as a social, collaborative enterprise and its connection to technology and society.
Yes, science is intensely social. Researchers build on each other's work. They share results through publications and conferences, and findings are subjected to peer review, where other experts in the field scrutinize the methods and conclusions before publication.
And results need to be reproducible.
Ideally, yes. Repetition of experiments by the original lab or replication by independent labs is crucial for confirming findings. This whole process acts as a kind of self-correcting mechanism for science.
Scientists also often rely on model organisms, right?
They do. Model organisms are species that are convenient to study in the lab. They might reproduce quickly, have simple genomes, or be easy to manipulate genetically. Think of the fruit fly Drosophila melanogaster, the nematode worm Caenorhabditis elegans, or the bacterium Escherichia coli.
Why study flies and worms if we care about humans?
Because of that fundamental eternity of life, rooted in evolution and the shared genetic code. The basic cellular processes, the functions of many genes, are often conserved across vast evolutionary distances. Discoveries made in a fruit fly about how cells divide or how genes control development often provide direct insights into those same processes in humans, including understanding diseases like cancer.
So model organisms are powerful tools because of shared ancestry. Now, what about the link between science and technology?
It's vital to distinguish them, though they are deeply intertwined. The goal of basic science is figuring out how the natural world works. The goal of technology is application, using that scientific knowledge for specific, often practical purposes.
Like the DNA example.
Exactly. Basic research in molecular biology uncovered the structure of DNA and how to sequence it. Science. That knowledge was then applied to develop DNA fingerprinting technology used in forensics. Technology. The Innocence Project, for instance, uses this technology, based on scientific understanding, to exonerate people wrongly convicted of crimes.
So they feed each other. Science enables new technologies, and new technologies allow scientists to ask new questions.
Absolutely. They are interdependent drivers of progress.
Finally, this progress, especially in biology and medicine, raises social and ethical issues.
Immensely so. First, the scientific enterprise itself benefits enormously from diversity. Different backgrounds, perspectives, and experiences lead to asking different questions and interpreting data in potentially new ways. A lack of diversity can genuinely hinder scientific progress.
And the technologies themselves raise ethical questions.
Profound ones. Think about rapid, cheap genome sequencing. It opens up amazing possibilities for personalized medicine, but also raises huge ethical questions. Who owns your genetic data? Should insurance companies or employers have access to it? How do we prevent genetic discrimination? How do we ensure equitable access to these powerful new therapies?
These aren't purely scientific questions.
Not at all. They involve societal values, politics, economics, and philosophy. Deciding how to use the powerful knowledge and technologies that science provides is a responsibility for all of society, not just scientists.
Well, this has been an incredible journey. We started with the sheer, almost overwhelming scale of biology. But now, hopefully you, the listener, have this robust framework to make sense of it all.
That's the goal. The 5 unifying themes. Organization, revealing life's hierarchy and emergent properties. Information, encoded in DNA and expressed through complex processes. Energy and matter, flowing and cycling to sustain life. Interactions, from molecular feedback to ecosystem dynamics and global climate.
And binding them all together, the core theme, evolution, explaining life's unity and diversity through descent, with modification and natural selection. Mastering these connections is really key to that professional level understanding.
These themes aren't just categories. They are the interwoven principles that explain life itself. They show us that life emerges from organized complexity, operates on precise information, is fueled by energy flow and matter cycles, and is constantly shaped by interactions within the context of its evolutionary history.
As a final thought to leave you with, something to really chew on, let's consider the sickle cell gene. In many parts of sub-Saharan Africa, carrying one copy of this gene provides significant protection against malaria, a deadly disease that's historically been very common there. However, inheriting two copies causes sickle cell disease, which is debilitating.
Now imagine a large population of people carrying this trait migrates from that high malaria region to a place where malaria is virtually non-existent, like the United States. How does the selective pressure change?
Well, the advantage of the gene protection against malaria disappears in the new environment. All that's left is the disadvantage, the risk of sickle cell disease if you inherit two copies.
So, over many, many generations in that new malaria-free environment, what would you predict would happen to the frequency of the sickle cell allele, the gene variant? And what does this scenario reveal about how the environment constantly edits the traits within a population, and why evolution isn't about progress towards some perfect ideal, but rather a continuous process of adaptation to current local conditions?
Something to think about as you apply this powerful five-theme framework to understand the living world around you. Thank you for joining us on the deep dive.